Today we will study about microbiology. In order to study infections, we need to know a little bit about microbiology and why it is important. So what our Ayurveda says about microbiology. Pratyaksham hi alpam, analpam, apratyaksham asti. It means प्रत्यक्ष ही अल्प है. जो प्रत्यक्ष है वो अल्प है. अल्प means it is less. अनल्पम अप्रत्यक्षम अस्ति. जो अप्रत्यक्ष है मतलब जो हमें दिखता नहीं है, the things which we cannot see, they are analpam. Analpam means they are in huge quantities, they are not less. यद यद आगमन अनुमान युक्ति अभिलुप्यते अभि उपलभ्यते. Right? So how do we see the aprathyaksh? Through agam. Agam means aptopadesh, through anuman and through yukti. So in Ayurveda also we believe that things which we cannot see, they are in huge quantity and they can be perceived either by the existing scriptures or by the aptopadesh, or they can be perceived by applying some yuktis or by making some guesses. So in Atharvaveda, two types of krimi are described, drishya and adrishya. Adrishya means which are invisible. And further, these krimis, they are also called as durnam and sunam. Sunam means non-pathogenic and durnam means pathogenic. Pathogenic means which causes pathogenesis, which causes some abnormality or disease. Also, Acharya Charaka has stated two types of krimis, and these two types of krimis are also pathogenic and non-pathogenic. So agantuj vyadhis are caused by krimis. Also, Acharya Sushruta has described many diseases like kushta, jwara, shosha, which are contagious and which spreads through direct contact or by using the contaminated objects. So in Ayurveda also there are various mentioning of microorganisms. So let us learn the modern developments. So in 1676, Antony von Leeuwenhoek, we already know this name, he developed a microscope. You can see the microscope, this microscope, this rat catching device like microscope. So the Antony von Leeuwenhoek, the great scientist, was called as father of microbiology because he used to observe minute things under his microscope. However, first recorded microscopic observation was made when Robert Hooke made a good microscope and started observing the molds, the fungi under it. Now let us see what is microbiology. We already know that the things around us, the living things around us, constitutes of plants and animals. We can very much see the plants and animals. We broadly categorize the living forms into plants and animals. So Aristotle, the father of medicine, he classified living organisms into plants and animals. Then in 1866, Ernest Haeckel, after the discovery of microscope, he classified another group of organisms. They were called as protists. They were neither called as plants or animals, but they were called in between to those plants and animals, and they were microscopic. Then we further had 2 kingdom classifications, 3 kingdom classifications, and ultimately we have 5 kingdom classifications by Robert Whittaker, which is more prevalent and which is more universally adopted. So this 5 kingdom classification we have already studied in 10th class or 11th class: Monera, Protista, Fungi, Plantae and Animalia. So now plants and animals everybody understands. We have seen plants, we have seen animals, humans are included in animals. So now there are certain microorganisms which are classified under Monera, Protista and Fungi. So what is Monera, what is Protista? Let us look into that. But before we look into Monera and Protista, let us first look into another word called as prokaryotes and eukaryotes. Now what is prokaryotes and what is eukaryotes? Basically these cells are classified under prokaryotes or either eukaryotes. So the prokaryotes were the previous cells or the archaea cells or the cells which came into existence first. So these prokaryotic cells, they were ancient cells, and they were having difference in their cell organelles. In fact, they were not having any cell organelles. All the membrane bound organelles were absent in them. The DNA was lying in the cytoplasm freely without any membrane. The ribosomes are also of 70S rather than of 80S. So the protein manufacturing machinery is different. And the eukaryotic cells have membrane bound organelles like nucleus, like mitochondria, like Golgi apparatus. So these cells which have membrane bound organelles, they were classified under eukaryotes, and although there were many other differences like ribosomes. And the previous cells, the ancient cells, they were put under the category of prokaryotes. So prokaryotes and eukaryotes are the first division that the scientists made once they got to know the internal structures of the microorganisms. So the Monera, they included the prokaryotes. The prokaryotes which were unicellular, and which were not having any organelle inside them, or not any membrane bound organelle. Similarly, the eukaryotic cells which were unicellular and some of the multicellular like some algae, those eukaryotic unicellular, mostly unicellular organisms were classified under protists. And the next category is fungi. Fungi, they were having characteristics of both plants and animals. Now, what is the peculiar about Monera and Protista, that apart from few exceptions, none of them they could photosynthesize, whereas Plantae can photosynthesize. So some of the characteristics in fungi, they are from plants, and some of the characteristics are from animals, right? We already know what fungi is. So Plantae, we already know plants and animals. So this 5-kingdom classification is just for your knowledge. So the prokaryotic cells, once again, the prokaryotic cells, the cells which do not have any cell membrane, which do not have any membrane bound organelles, those moneras, right, those prokaryotic cells were called as moneras, and those moneras or those prokaryotic cells contains the bacteria which we are now concerned of. The protists, which are eukaryotic cells, example protozoa, example malaria, the protozoa which causes malaria. So these protists are eukaryotic cells, whereas bacteria are prokaryotic cells. So let us talk about the thing which we are concerned of, infection. So the infection in human beings is mostly caused due to these agents, these microorganisms. Prions is not a microorganism, it is just a protein. It is just a protein which is, you can say it is a mismanufactured proteins produced by either by the body itself or by some other body, by some other animal which comes into the human being, and it deranges other proteins also and causes certain diseases. One example of prions is mad cow disease, which comes by eating cows, right? From cows, this deranged proteins, basically proteins are helical structures. They are alpha helical structures, just like DNA, proteins are also helical. But when this structure, this helical structure changes into some other structure, say a step ladder structure or say a Z type structure, that is called as beta stranded structure. So when this alpha helical structure changes into beta structures, then these proteins are now dangerous, they causes diseases, and these are called as prions. So prions are not microorganisms, they are just proteins which causes diseases. Example, mad cow disease. Then we know what is a virus, we know what is a bacteria. Viruses are also not microorganisms, they are also not organisms. We don't consider under them organisms because they don't have a metabolic function, because they don't produces metabolism, or they don't do metabolism on their own. Basically, what is a virus? Virus is a genetic material surrounded by a membrane, right? Surrounded by a cellular membrane, that's it. So viruses don't have ribosomes, viruses don't have mitochondria, viruses don't have any cell organelle, right? So they are not considered as living, they are considered as dead. Whereas bacteria are living organisms. That means they don't need humans to survive. They can survive on their own. They can survive in the soil, they can survive in the hot water springs, they can survive in the extreme temperatures, they can survive, even in the mouth there are so many bacteria, and the bacteria are very picus. the majority of the earth biomass, the living mass, is made by bacteria. And even our body contains trillions of bacteria. Even our mouth contains bacteria more than the number of people on this planet. So bacteria, they are in huge quantity. Even one handful of soil contains more bacteria than the total population of the planet, total human population of the planet. So bacteria constitute the major cellular biomass of the planet. Plus, bacteria also produces more oxygen than plants combined together. So the major producer of oxygen on our planet is bacteria. Then we have protozoas, then we have helminths. Now protozoas they are unicellular organisms, protozoas, but they are eukaryotic organisms. If you see the bacteria are prokaryotic microorganisms, whereas protozoas are unicellular but eukaryotic microorganisms. Now, why I am stressing on prokaryotes and eukaryotes? Because prokaryotes and eukaryotes have different cellular machinery. So when they have different cellular machinery, we will be requiring different agents to kill them. For example, if we know that antibiotics are used for bacteria, but those antibiotics cannot be used for protozoa, because antibiotics works for prokaryotic organisms, not for eukaryotic organisms. Or the antibiotics won't work for virus, or the antibiotics won't work for helminths, like that. So we need different chemicals, or we need different life-killing chemicals for different organisms. So then we have helminths. Helminths are the worms, the tapeworms, the hookworms. And then we have arthropods like insects, or which causes infection, or mites, or ticks, or your lice, head lice, louse, right? All these things, they comes under arthropods. So now we will start with some of the few basic definitions. Infection. So I'll just skip these definitions because they are self-explainable, nothing much to explain in them. So you know what is infection, right? Infection is any organism which causes disease, as simple as that, right? Which causes disease. Then how do the infection transmits from one person to another, right? We'll study about that. So infection might travel from one person to another. It might travel from an infected person to a healthy person, or it might travel from an infected animal to a healthy animal. Now, what are the types of infections? The colonization. Now, certain bacteria, they just colonize the body. They colonize the body, and sometimes they don't produce any infection. They're just living there. They are just using the house as rent, house on rent, right? They're not producing any trouble. Now then we have subclinical infections. Subclinical infections they are producing some infections, but the symptoms are not fully blown, or symptoms are not present. So we call such situations as subclinical. Always subclinical represents the clinical features are not manifested. And then we have symptomatic infections. That means the symptoms are very much visible and the infection is full blown. Then we have some latency period and incubation period. We already know these terms. Incubation period is the time between infection and the onset of symptoms. Basically, the bacteria the microorganism is preparing itself or multiplying itself enough to produce the symptoms. And latency period is a little different than incubation period. Why I have put these two terms? So that you can differentiate between latency and incubation. The time between infection and the onset of infectiousness, right? That is basically when the virus, suppose we have already studied hepatitis B virus and hepatitis C virus. Sometimes these viruses, they just set into your body, or they are just inside your body for a very long time, right, without producing any infection. So that time period when the virus is silently just sitting around in your body, that is called as latency period. We will study about this latency period when we go into certain conditions like syphilis, or we will go into certain conditions like some other diseases. Then definitions. Disease, right? You know what is a disease? Infectious disease, hardly it makes a difference. Then pathogenicity and virulence. Pathogenicity is the ability of a microbe to cause a disease, right? And virulence is the degree of pathogenicity in a microorganism. This term is often used to describe or compare strains within a species, right? That means virulence means how much effective it is, right? How much virulent it is, how much it can cause the problem. For example, if I take one strain of bacteria and other strain of bacteria, if I compare both the strains of bacteria, that how much effective they are to produce disease, that is called as virulence. Then we have acute infections and chronic infections. Acute means sudden, chronic means delayed onset and slow progression. Primary infection, secondary infection. Primary infection means if a person is primarily infected with some organism, right? Secondary infection means if a person is having, say, a viral infection, and after that viral infection he suddenly got another bacterial infection, then that is called as, the bacterial infection is called as secondary infection. The primary infection makes place, or the primary infection depresses the immunity so that the secondary infection can jump on. Then we have localized infection versus systemic infection. Localized means it is particular to a region. For example, cellulitis. Sometimes we get a boil over our body part, that is a localized infection. And for example, if your eye is infected, that is a localized infection. Systemic infection means which involve the complete system. For example, fever. Or for example, malaria. Malaria is a systemic infection. Clinical infection versus subclinical infection. Clinical infection with obvious observable and detectable symptoms, right, which is clinically manifested. Subclinical again means which is the clinical features are not full blown, or clinical features are not there. Opportunistic infections. Now, certain microorganisms, they are residing in our body, and some of them, they are even pathogens, but they are not producing any disease. They are just staying there. But as soon as the immunity of the body goes down, say because of malnourishment or because of some other disease, for example diabetes, then these infections, these microorganisms, they start causing infections. Those are called as opportunistic infections. Then certain other definitions like epidemiology, right? What is epidemiology? Epidemiology is the study of transmission of disease. So we always study first epidemiology in a disease. After the definition, epidemiology is how the disease is transmitted from one person to other. Then communicable disease, right? Disease which can be transmitted from one person to another. Contagious disease, right? Contagious means which can easily spread. And then non-communicable disease, right? A disease that is not directly spread from one person to another. Then endemic diseases, epidemic diseases, and pandemic diseases. Now, endemic means when a disease is present only in certain percentage of a population, whereas epidemic means a disease which is present in big population. So what you can say, we can understand this by an example. Suppose if a district of a state is getting affected by a disease, we can call that as endemic disease. Or if a village is affected by some disease, say cholera, right, say dysentery. So if all the village people of one village are affected by a disease, we can call that as endemic. Epidemic means if the complete state is, or complete country is having that disease. Pandemic means global, just like coronavirus these days. Then we have reservoir of infections. Reservoir means the infectious agents lives in a particular source, right? What is the source of, for example, for this coronavirus, there was a organism called as, sometimes it is called as bat or pangolin, right? So these are reservoirs of infections. The infection stays in such organisms, and that infection transmits from there to the humans. Then we have carriers. Carriers are people, or they are individuals who don't have any symptoms, right, but they are highly infectious. They can spread the infections around them. Then we have fomites. Fomites are they are the agents of intermediate transmission, means they just carry the infection. They are not affected by the infection, they just carry the infection. For example, the plague. In plague, a rat carries a flea which contains the bacteria, right, which contains the bacteria Yersinia pestis. So that fomite, that rat is called as a fomite. It is an indirect transmission of an infectious agent. Then we have disease transmission, how the diseases are transmitted. So we have direct and we have indirect mechanisms. Direct means direct from person to person. For example, direct skin contact, or direct if some aerosols, right? Somebody sneezes and from the sneeze the virus goes out or the bacteria goes out. From mother to fetus, that is a direct contact. Direct contact means when a person is coming close to person. Indirect means, for example, food, waterborne, fomites, or and through animals, right? When the persons, when there is no direct contact between the people. Then we have animal vectors. Now, animal vectors can again be of mechanical and biological. Biological means the transmission of malaria. For example, transmission of malaria, right, is a biological animal vector. The infectious agent lives inside the animal's host and completes its life cycle, or it infects basically the animal host as well. That is called as biological animal vector. Whereas mechanical animal vector, mechanical means only it is using it as a vehicle. So the infectious agent is physically transmitted by the animal vector, but it does not infect the animal, right? For example, transmission of bacteria sticking to the feet of flies. Then we have modes of transmissions. Direct transmission, indirect transmission, right? So we can divide the direct and indirect transmission. We can easily form these things. Direct contact, right? We have already studied direct contact, percutaneous, through skin, through perinatal, through the mother to the unborn child, and through sexual, right? Droplet spread, right, through the sneezes. Indirect transmission, right? Indirect means when the persons are not in contact. Airborne, vehicle-borne, right? Airborne means there is some microorganism floating in the air, some virus floating in the air, and it goes into your system. Or vehicle-borne, for example, from the soil, contaminated water, contaminated air. Then vector-borne, right? For example, mosquito, right? From mosquito, so the organism transmits from one person to the another, right? Then we have a suffix called emia. We already know this. Emia means in the blood. So bacteremia means bacteria in the blood. Viremia means virus in the blood. Fungemia means fungus in the blood. Then we have itis. Itis means inflammation. So pharyngitis means inflammation of pharynx. Endocarditis means inflammation of heart, endocardium. And then we have gastroenteritis, right? Enteritis, right? Gastro means stomach, enteritis means the intestine. Then development of infection. Development of infection, we have incubation period. We already know what is incubation period. Incubation period is the time period when the organism gets into the body, but it does not produce any virulence, it does not produce any pathogenicity, and to the time period when it produces some clinical symptoms, right? So incubation period, the organism will not produce any clinical symptoms. Then we have prodromal period, the Purupavastha. In these symptoms, the symptoms are not very specific, they are not very clear, and the person says, I am ill, or I am uncomfortable, I am discomfortable. Then we have acute period when the infection is fully blown. Then development of infection, clinical signs and symptoms, local signs and systemic signs. Local signs, now locally, what can happen? Suppose if I am having an infection over my hand, inflammation, purulence, right? Purulence exudates means pus coming out of that. Tissue necrosis, the tissue might die, become black. Lymphadenopathy, right? So these are local signs. Then systemic signs, fever, fatigue, headache, nausea, right? They are systemic features. The generalized stages of infection, the pathogen enters, it colonizes a particular site, right? For example, some viruses, they go to the liver cells and they colonize the liver. Some viruses, they go to the respiratory tract, they colonize the respiratory tract, right? Then we have an incubation period when the microorganism is preparing itself to attack. Then the prodromal symptoms, right? The initial symptoms or the unspecific symptoms. Then we have invasive period, right? Invasive period means when we have acute infection or when we have the actual clinical features. Diagnostics. Now, how these microorganisms are diagnosed? So there are many methods. I have not summed all of them. However, I have summed few of them, right? So the most previous and easy method are stains. We stain the bacteria, right? We will study about these stains. Then we culture the bacteria, right? We take the bacteria, put it in a growing medium, for example agar agar, and we put that bacteria into that, and we allow the bacteria to grow, and then we check for that bacteria, or we do certain testing with that bacteria regarding to some antibiotics, or regarding to some staining, or regarding whatever. So we culture the bacteria, we basically proliferate the bacteria, and then we do the testing so that we can recognize the species of that bacteria. Then we have dark field microscopy or fluorescent microscopies. We might talk about them later. Then we have blood tests, right? So blood test in a general, in a nutshell, we can summarize things, although there are many exceptions. Bacteria, leukocytosis. When the WBC levels are up, we say it is a bacterial infection. Viruses, leukopenia, right? Right? When the leukocytes are less, when the WBCs are less, we call that it is a viral effect, viral infection. Then if the neutrophils are increased, we say it is an acute. If the lymphocytes are increased, we say it is a chronic infection. These are general things, not very specific to all. Then we have another test, polymerase chain reactions, reverse transcriptase, where we reverse transcript the DNA of the virus, and we make multiple copies of that DNA virus, and we do the testing. For example, PCR test is being done for coronavirus to detect the coronavirus, the COVID-19. Then we have antibodies and antigen testing. We can specifically test the antigens and antibodies. For example, we have already studied the hepatitis, right? The hepatitis, we do the antigens and antibody testing. So let us have a break, and then we will start the bacterial infections.